My name is Leah and I'm a high school junior. I've always loved GETT because the speakers have inspired me so much over the past few years. My favorite subjects are math, computer science, and animation. I hope you enjoy the video and happy 20th anniversary, GETT!
Hi, I'm Glenda, and I'm a scientist at Janssen Pharmaceuticals. Did you ever ask yourself, what does it take to make a new medicine?
Hi, my name is Sabrina Sirez DeLong, and I'm a scientist working at Janssen Pharmaceuticals. I started my scientific career by completing an undergraduate degree in medical biochemistry and later went on to complete my PhD in immunology, followed by postdoctoral training. At Janssen Pharmaceuticals, we work to discover new drugs to treat certain diseases. The journey to discover these new drugs starts and finishes with the patient. In our body, some proteins may not function correctly in certain diseases. Once these proteins have been identified, a team of researchers are then put in charge to discover new molecules that can bind to the proteins to uniquely regulate their function. Today, you will be part of a team to discover a new medicine.
Now you're part of the team, and the materials that you will need to start your new molecule are clay, marshmallows, or other materials that it will represent your molecule. Then you will need straw or sticks that will represent the bond that will make the molecules join. And then you will need scissors in case the sticks are too long. Then you will need a cup or an empty jar that will represent your protein of interest. Have fun building your new molecule.
Hello, my name is Hui Huang. I'm a senior principal scientist at Janssen Pharmaceuticals, and I'm a part of discovery chemistry team at Springhouse, Pennsylvania. I attended Nanjing University in China for my bachelor's degree. In 1995, I came to the United States and attended graduate school at Northwestern University. In 2000, I graduated with a PhD in organic chemistry. After graduation, I worked for a small biotech company for 3 years, then joined Janssen Pharmaceuticals in 2003. Today, you will be a part of the medicinal chemistry team who works with the biologist to find a molecule that interacts with the disease-causing protein target and is safe to treat patients. It is a long journey for drug discovery. Normally, it takes 10 to 12 years from discovery to market a drug. As a chemist, you will use computers and other technologies to see what the protein looks like closely. Then you either design new molecules that bind to the target proteins, or you find and modify pre-existing molecules as a place to start your work to make more effective and safe so-called drug-like molecules. Then you will make the molecule in the lab using chemistry reactions to put together smaller molecules and assemble the molecule you designed. It is very cool to create new molecules that have never been made before in the lab.
So here is the tricky part. The molecule that you construct needs to fit into the cup or anything that you choose that will represent your protein of interest in order to improve your patient's health.
Hello, my name is Fanny Guerra Bonilla. I'm a senior associate scientist at Janssen R&D. I have a pharmacy degree obtained at the University of Panama, where I studied for 4 years. I also have a master's degree in pharmaceutical sciences. In this step, you will learn that your molecule must bind to the protein with precise fit. Typically, a molecule that binds to the protein of interest is created. Then the researchers may modify it or create and test new molecules in a process that looks to optimize the properties that are important for a successful drug candidate. Those properties include pharmacokinetics, or how the body interacts with the molecule, the efficacy, where we will observe if the drug has the effects that we expect, and the safety, where we will learn about adverse effects or toxic effects. All this process is called preclinical development, and it may take many years to complete. For this activity, your molecule shouldn't fit too tight to the cup that will represent the toxic effect, or cannot be too loose. That will mean that the therapeutic effect is not achieved.
So now, were you able to finish your molecule? Were you able to put your molecule into your protein of interest and easily take it in and out? If your answer is yes, then you have a drug candidate that will move to the next stage, the clinical trials. If not, you need to keep working in your molecule to make it a successful drug that can move to the next stages. Good luck.
Hi, my name is Ademi, and I'm a biomarker researcher. I've always been interested in science and decided to go to college for a 4-year degree or a bachelor's in science in biochemistry. After finishing my bachelor's in science in biochemistry, I decided to pursue graduate school. And so I went to get my PhD in pharmacology, and that took about 6 years to do. After finishing my PhD study, I wanted to get a little bit more training and worked as a postdoctoral researcher where I studied the disease melanoma. After finishing my postdoctoral training, I decided to join the pharmaceutical industry as a biomarker researcher, and that's the work I've been doing for the past 10 years. So as a biomarker researcher, our goal is to understand who are the right patients to give the drug to. And how we do this is in the context of clinical trials. So up until now, we've heard a lot about molecules and what it takes to get them ready to go into the clinic. And the clinic is the first time that the drug is delivered to a patient. And so it's really important to make sure the drug is safe and to really understand that we're finding the right patients to give it to. And so my job as a biomarker researcher is to understand the characteristics of the diseases we're studying and to find the right patients to give the drugs based on markers in their tumor or biomarkers in their tumor. So we use these clinical trials to understand how effective a drug is. Is it working? Is it working the right way it's supposed to be working? And to do that, to be able to answer those questions, we collect data. And we analyze that data to understand if the drug is working the way it's supposed to, and to understand if there's ways that we can make it work better.
I hope you have fun being part of our team today. In real life, making a new molecule can take us several years, but the satisfaction of knowing that we make a difference in a patient's life is unmeasurable. Before I joined Janssen, I completed a bachelor degree in biochemistry, and then I pursued a grad school doing a master in marine biotechnology. I hope we encourage you or exciting you about pursuing a career in STEM. Have a great day.